I don't even know. I'm sorry. That question sucks. I don't know where that came from. That was like a 106 trick question. Heritable traits are applicable to, it just, yeah, I don't know what the test bank says. I would say it would be, by definition, an individual. Because, right, because you can't, did you get it right? Oh, you don't know because it was 100. I'm like, you got 100% and anybody had that question. Tell me what you put down. That was the answer? Yeah. Because for me, being a molecular geneticist, I would say the heritability is only from parent to offspring. But if you think like an ecologist, heritability as far as natural selection goes occurs in a population. We always talk about evolution occurs over time, change in trait frequencies in a population. So a population geneticist would say across the entire population, and a molecular geneticist would say an individual. Because really, the trait is passed from me to the child, not to another child in the population, right? But the overall population's trait frequencies can change based on survival of the fittest and all of that crap. So if you got that question and you got it wrong, let me know and I'll give you a free point. So just send me an email and I'll add a point somewhere in a bonus section, because that one I meant to get rid of because it's always a problem. I don't even know where it came from. Some of these are just test bank shit I pull in and looks like a good question and then it sucks. If you got it wrong, like you're missing a point because you had that question, just send me an email, I'll give you extra points. Because that one sucks, and that was a mistake.
I got it wrong, but I think the problem is that just came from 106.
Right.
And the way that they had it is just like the individual is heritability.
Right.
When the population is natural selection.
Exactly. And as a molecular, I'd say, but the term measures of heritability is vague, right? What the hell does that mean anyways? So yeah, I apologize for that. That sucks. Yeah, sorry. You got it. Okay, guys, are we ready? Are we ready for another day? This really is supposed to, it's supposed to be the other little video. Let's see if it works. I just reloaded it. Oh, it's going to work. So this is the cleavage. This is cleavage. Are you ready? We're going to watch porn today. Yes! Okay, hands on top. Everybody's hands. No hands on your lap, please. Oh my God, it's cleavage. Okay, so this was a zygote. Again, we're under time lapse. Right? One single cell fertilized to mitosis, 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 mitosis up the wazoo. Is that crazy? Okay, let's watch it again. So one single cell, which you can barely see, it goes so fast, and then divides in half, and each in half, and each in half, and so on and so on and so on. See? Yeah, it's definitely time lapse. So really, this happens over the course of about 7 or 8 days. Okay? So, we're supposed to be, yeah. No. So that's a great question. And that's one of the huge misconceptions that I didn't even pay much attention to until I saw this video, 8 years ago. So my whole life I had this, wrong misconception. Remember we saw that picture of the egg and it was ginormous compared to the little tiny sperm, right? Dr. Stein always draws when we used to be able to use a chalkboard and a big, huge circle over the whole chalkboard and then like a little dot for the sperm, right? Everything in that egg is what we need to start mitosis and to get to that huge ball of hundreds and hundreds and hundreds of cells. Because fertilization happens somewhere usually in the fallopian tube, right? We get ovulation, the egg starts to travel, the sperm finds it somewhere in the fallopian tube, fertilization happens, and then mitosis, 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 as the egg's traveling down the fallopian tube trying to get into the uterus. It takes about 7 days to get there. And the uterus is all full of fleshy, good tissues, right? Highly vascularized. that ball of cells sticks in the goo, right, and then continues development. Goes through blastula, gastrula, right, and then at about another 7 or 8 days, the very beginning of the placenta, right? It's starting to form as soon as it goes through gastrulation or blastulation at the blastula stage, but isn't really formed to get really good connection and gas exchange and nutrient exchange until another 7 days. So 2 weeks from the time of fertilization. Okay? And so that's the point. The placenta is what's sending out that hormone, that protein that the pregnancy test tests for. Okay? And so that's why generally you can't detect whether someone's pregnant or not until 2 weeks after fertilization. What would be the first day of your period if you weren't pregnant? Okay, because it's trying to detect that hormone that's coming off of the placenta, right, the implantation. So there's lots of fertilization events that happen that don't ever implant correctly, or they implant but then don't make a good placenta and go away. And none of those anybody knows happen, right? So about 2/3 of all fertilization events go away without making, without resulting in a live birth. So during that entire time, there's no additional nutrients, there's no additional oxygen, there's no way to dump carbon dioxide, yet we're mitosing up the wazoo using tons of energy, right? So all of that had to be stored in the egg. So it's the exact same size as the original egg until implantation, we get the stupid placenta going, we get the gas exchange and the nutrients, then it can start expanding inside. Right? So it doesn't zoom out. This is exactly what it looks like. It starts the size of that egg, and then, right, G1, we don't grow like we did, we talked about mitosis. G1 has to get bigger, right? It has to be ready by G2, it has to be twice as big, so when it splits in half, they're the same size, right? In this case, there's none of that. We're ripping through G1, right? We're not growing in size, we're replicating DNA, we whip through G2, mitosis. Right? Because you can't. A cell can't grow in size without input of energy. Right? It has that finite amount, everything it needs to get to that size, to stick into the uterus, to implant, to start building the placenta. Right? So it's a very anoxic, low oxygen situation. So if there's low oxygen, this is going to be a question to remember from the past, as a review for the final. If there's low oxygen, what kind of metabolism are these cells doing to generate, right, some anaerobic, or at least just, right, glycolysis, right? They can do glycolysis under low oxygen, you don't need that, and then some fermentation to regenerate NAD to keep doing glycolysis. Okay? So the egg does store tons of all those things needed to do glycolysis, lots of sugar, right, fat, I'm sure it's even, all the stuff that it needs to go, but mostly sugars, mostly carbs, because that drives glycolysis. Okay? So it's not really going to switch to real cellular respiration until we're hooked up with the placenta getting that oxygen. And now we have an oxygen supply, now we can go down Krebs and electron transport chain. Right? And then get the big mother load of ATP. Then we can start growing. Each individual cell can grow in size as they're doing mitosis, making more of themselves. Just a little fun fact. Okay, that was cleavage. Woohoo! Okay, the last thing we're talking about for genetics is all the stuff that we've talked about so far, except for the hitchhiker's thumb and all those goodies, tongue rolling. Most of the other things are all non-human traits, okay? There's only a list of, 20 or 30 traits in human beings that are one gene trait. Most of the others are either quantitative quantitative, where they go like the bell curve, like height or weight or skin color, eye color, hair color, right? Or they're traits that we can't physically see very well. Okay? Even the ones that are one gene traits, how do we know in humans whether they're recessive or dominant? Way back when, before we determined they were recessive or dominant. We can't really do a parental cross, right? We can't really breed some people to make sure they're true breeding for the straight, forehead, or true breeding for the widow's peak, and then go, hey, now you two have to have sex to see what the kids look like, right? We don't do controlled breeding for human beings, but we can look at family histories. And so a pedigree is just your family history. And then we can trace these traits to try to figure out whether they're dominant or recessive. So when we look at, especially human diseases, that's how we had to figure out how they were passed. Cystic fibrosis we now know is a recessive trait, right? Huntington's we know is a dominant trait. Hemophilia is a sex-linked recessive trait. All of these were done by just looking at who in a family had these diseases. So it had to be a pretty big extended family where we know lots of cousins and offspring, because otherwise you can't follow it if you just know parents and child. Even grandparents, parents, and child, not necessarily straightforward. Yeah? So environmental factors definitely show up, and so there's anomalies on some of these family trees, and again, the bigger they are, the easier it is to say, okay, this person should have shown this phenotype, but didn't, we don't know why, but everything else falls into a normal recessive, trait, or a normal dominant trait, but occasionally, and then that's the reason why there's lots of diseases out there that we still don't know, because they don't follow a classic Mendelian inheritance, right? They pop up here, they pop up there, a sibling that, if you do the math and the statistics, is a big enough family, should have shown up, doesn't, right? And when in Genetics 360, we talk about all the environmental things and other ways crazy stuff can happen. But a pretty straightforward disease like cystic fibrosis, right, is gonna show up statistically when it's supposed to, especially in big families. So it was much easier to follow pedigrees, A, in countries that have lots of children, or B, my great-grandparents each had 13 siblings or some crazy shit like that, right, to follow these kind of things. And then a lot of them were followed in royal families because there's lots of them. They kept good notes on who's related to whom because they were only allowed to marry other royalty. Right? If you're only allowed to marry other royalty, your parents need to keep track of who's royalty so they know who you're allowed to marry. And in those cases, they're very inbred because you're only allowed to marry royalty. If royalty started as one family, right, you're marrying your cousin or your brother, right? So if you look at the royal family's pedigrees, incest is best, right? And that's why lots of bizarre recessive diseases show up. If one person in the family was a carrier, used that term on the quiz yesterday even though I didn't talk about it, has an allele for a recessive disease, looks phenotypically normal because they have one dominant, one recessive, but they're carrying that allele. They marry somebody else who's a carrier, right? So that's a heterozygote times heterozygote. If we do the Punnett square, 1/4 of those are going to be homozygous recessive. So if those two marry, 1/4 of their children should show the trait be sick, have hemophilia, right? Or cystic fibrosis or whatever the other trait is. Hemophilia happens to be sex-linked, so the boys all show up with it, but the girls don't, right? And then they're all intermarrying, so there's lots of carriers. The other kids who look phenotypically normal, half of them will be carriers, only 1/4 of them will be homozygous normal, right? So then if that carrier has to marry their cousin who's a carrier, 1/4 of their kids have it, half of them are carriers, and it just takes over this whole thing. So what I want you to know for 107, because we do pedigree analysis up the wazoo in 360, and I just want you guys to understand the basics here, is why we do it, right? Because we need to look at family history to determine how something is inherited in human beings where we can't do controlled mating, right? And that we use these and what the symbols look like. So that's just a memorized thing. There'll be nice, easy questions for the exam. We're always needing more easy questions on the exam. And then when you go in to take genetics, you already know the setup of a pedigree and what it's pointed. Yeah. No, we're going to know the simple stuff. Right, so we definitely need to know the male and female. So males are always squares, females are always circles. The mating line, which, you know, in the good old days they called the marriage line. Oh, wasn't that cute? Because, you know, people don't have sex unless they're married. Right, yeah, romantic, yeah. Wasn't it romantic? And so this would be a setup of what it looks like. The line comes down from the marriage line, the sex line, and then how many kids, right, they have from that. This we don't really care about. I mean, it's nice to know if you see it, oh, look, there's some twins, but it doesn't, you know, we don't care about, you know, how those guys work. And same with this stuff. The only thing is the triangle just means you don't know what that was. Like in a family, you know, a family member says, oh, my sister had 7 kids. I know she had 3 boys and 3 girls, but I can't remember what that baby was because I haven't talked to her or whatever, you know. So, or way back, you can't tell their name was some androgynous name. That's all you know about it from a tombstone, right, when you're doing your history. So you just put a triangle, you don't know. So for anything that's an autosomal disease, it doesn't matter. If it's a sex link, then it does matter if it was a boy or girl and whether they had the disease or not. But if you don't know, you don't know, so you just put a triangle. Yeah?
When it says parents and children and it's averages for the order of birth, that goes like left to right?
Yeah, it just usually goes left to right in order of birth. And on a real pedigree, say, I don't know, I can't remember what this one looks like. Yeah, they're numbered, right? So each individual's numbered. So generation 1, they list in Roman numerals. So if somebody asks you about, you know, the female number 1 in generation 1, then you know, or female number 3 in generation 2 is this person. Right, generation 3, and then they list them so that, you know, you can ask questions about them. And yeah, the birth order is usually left to right. Yeah. If they know it.
For example, like you're doing for the parents on top and then it goes down to the children. Would the 1 through 7 be the children, or would it be the 1 to start as like a family?
Yeah, so that's a good question. So really the kids follow this line, and then the people who marry in don't have the line. So that would be the spouse outside of the family. And normally it's just black and white. I don't know why this figure had to. So the red would be the dark, filled in. Those are the people who have the trait. You know, if we're talking about a disease, they're sick. They have it, they're sick, whatever. Huntington, they have Huntington disease. Or they don't have to be sick, but they have the trait. They have the straight forehead, or they have the tongue rolling trait, or, you know, whatever it is you're talking about. And then the open ones, the unfilled, or here, yellow, are the ones that are phenotypically normal. So yeah, that tells you who's, and this little clause is really just for 360. This is a slide from my class in 360. People who marry into a family, if there's no reason to suspect they're a carrier, we assume they're not. So if you have Huntington disease in your family, right, and you meet some random person at a bar, it's likely they're not a carrier. Huntington is a rare disease. Cystic fibrosis is a rare disease. Anything that's rare, it's unlikely somebody you bump into online or whatever is going to be a carrier. Now, if, you know, in 360 they say, if I tell you that, I'm not going to do that to you guys, if I tell you they met at a support group for cystic fibrosis, a carrier, right? But you can look at the children to determine it. But in here, we're not going to actually do any analysis because we're going to do that like crazy in 360 and we're out of time. So I really just want you guys to know what the point is, right? The affected guys are the filled in. Usually if we know something, they might color them heterozygous. A lot of times we don't know, right? If they happen to be dead, there's a line, but I don't really care about that. And we don't really care about this either, okay? This means they're marrying somebody in their family, first cousin or closer. Right? And so that immediately tells you something about this is an inbred family tree. It's likely to have some sort of rare recessive disease going on in there. So it's kind of a giveaway. And then this one is the one that just talks about the generations and how they're set up. So really just the setup of the pedigrees and what their overall point is. But no analysis. I won't give you a pedigree and say, is this a dominant disease or a recessive disease? But I might say, is the, what is the sex and are they sick or not of, individual 2-4, right? So Roman numeral 2, second generation, number 4. If it's a closed circle, you'd say female affected. So easy stuff like that. Right? We need some easy questions. What day is it? Thursday? Okay. Yes. If they're within the family, we have to assume they could be a carrier. And then you can tell by the kid, like if two normal phenotype people have a sick child, they both had to be carriers, right? They both had to give up a little r, a little r. But we're not going to worry about this for this class. But for the future. So I'm not going to muddle your heads with that. Oh, crap, I don't have... I don't have my lecture ready. Okay, take a break, talk amongst yourselves. Or think of questions. And Dropbox isn't letting me. See, I should have had this all set up. But I don't. Alright, I need Wi-Fi. Oh, it says I have Wi-Fi. Okay. Jacked up. It should be Tuesday the 19th. Is that a Tuesday? Oh, yeah, it's the 19th. Sorry. All right. Do I have what I need here? Okay. I cannot get into the box again. Oh. That's a good question. So for the final exam, in general, we don't use the same exact question that we used on the midterm. The midterms in general are more specific than the final. In some cases, like for the organelle chapter, we're going to have to ask you about one or two of the organelles, right? There's no like overlying theme which you absolutely have to know. So we'll ask a function of one of the organelles. It'll be similar to one of the midterm questions. Right. So we won't pick another specific area to cover. So like so for me, when you look at a lecture and there's like maybe 2 or 3 questions per lecture, some of the final is going to be just the last stuff, right? Just like a regular midterm. And then the cumulative questions will be like, for the monohybrid genetics, what main thing do I want you to know? Well, you have to be able to do a Punnett square. So there's going to be one just stupid, the father's heterozygous and mother's homozygous, and like, well, what are all the kids? What's the proportion? One of those, one problem, maybe something about homozygous, heterozygous, some main, without picking anything super specific. I don't think so. Usually the cumulative stuff of the final is multiple choice, but I'll ask her to make sure for that section how she wants to do it. Yeah. Okay. Okay, so since this is an old lecture, I can't. Okay, are we ready? Are we ready for replication? I'm ready. I'm finally ready. Uh-oh. Are we ready? No, we're not ready. Okay, let's get ready. Look, I got a new chord. Look at how much I can wander now. Isn't that exciting? Ooh. Okay, so let's do this right now. Let's not do this. No. Let's not do this. And we're not doing this. Oh, look, we're not doing this. As much as I love this experiment, we do it in 360, and we don't have time to do it now. Okay, this isn't going to link, so we're just going to... All right, let's just do something. How about that? Let's do something. Let's do... Nope. Yeah, no Hershey Chase. As much as I love viruses in a blender, it ends up taking the whole lecture. We have too much shit to cover. Yeah, it's the phage experiment. Yep. Which again, I love, but I get to do it in 360, so. Okay, so we're going to just watch a little review because we're talking about DNA, and this is just a lame little video about DNA base pairing, right? And so we need to make sure we know all of that stuff from chapter whatever that was, 4, 3, whatever, nucleotides and all that crap, so that we can start with replication knowing, I'm going to talk about it, but without going through it ad nauseum. Ooh, let's see if we can listen to their cheesy talking now that we have, where's my... Sometimes the audio in here is really bad. It usually is. Can you hear that? Yeah, okay. If it blasts you out, sorry. It's either, like, you can't hear it at all, or it's going to make me pee in my pants.
DNA is composed of 2 strands of nucleotides, wound around one another in a double helix.
Riveting.
The outer longitudinal structure consists of alternating sugars and phosphate groups. Paired nitrogenous bases are in the interior of the molecule. If we unwind the molecule, its structure resembles a ladder with phosphates and sugars forming the sides. The rungs are the nitrogenous bases of nucleotides on opposite sides of the ladder. There are 4 different bases, and only certain pairs are possible in forming the rungs. Adenine is always opposite thymine, and guanine is always opposite cytosine. These complementary pairs of nitrogenous bases lie next to one another because their sugar-phosphate chains run in opposite directions. This anti-parallel orientation of the 2 nucleotide strands brings the bases into position for hydrogen bond formation. The sides of the ladder are twisted in a spiral fashion and make a complete turn every 10 base pairs.
Is that guy not the most riveting speaker ever? replication, and then we're going to walk through all the steps on this. There's also the little tutorials that I put in there where it's me drawing and telling you riveting things. So make sure you watch that too. Okay, are we ready for him again? Try it.
The rule of base pairing helps explain how DNA is replicated prior to cell division. Enzymes unzip the DNA by breaking the hydrogen bonds between the base pairs. The unpaired bases are now free to bind with other nucleotides with the appropriate complementary bases. The enzyme primase begins the process by synthesizing short primers of RNA nucleotides complementary to the unpaired DNA. DNA polymerase now attaches DNA nucleotides to one end of the growing complementary strand of nucleotides. Replication proceeds continuously along one strand, called the leading strand, which is shown here on the left. The process occurs in separate short segments called Okazaki fragments next to the other, or lagging strand on the right. This difference is due to the fact that DNA polymerase can only add new nucleotides to the 3' end of a nucleotide strand. A primer begins any new strand, including each Okazaki fragment. An enzyme replaces the RNA primer with DNA nucleotides. Then an enzyme called DNA ligase binds the fragments to one another. There are now 2 DNA molecules. Each consists of an original nucleotide strand next to a new complementary strand. The 2 molecules are identical to each other.
Those are the sister chromatids, right? And then somewhere they're actually connected with their centromere. So this is occurring in S phase. Right? We talked about an interphase, right? That's what actually happens in S phase. So some of the things are not exactly correct that they showed. Most of the overall stuff is okay. There's a few things that are wrong molecularly, but for us, we don't really care. I hope. Okay. So when they showed us in that little video where it started, right, where the proteins came in and opened the double strand immediately and broke hydrogen bonds, they called it unzipping, which is, I don't like that term because it's really just breaking those hydrogen bonds and opening up that double helix. And again, transiently, because DNA always needs to be double-stranded, and as it turns out, any nucleic acids that are near each other that can complementary base pair will, by the force of chemistry, right? Hydrogen bonding. If you put a strand here and a strand here and it's anti-parallel and it's the same sequence, it's going to hydrogen bond. So as it turns out, when it really opens, it has to be held open. So there's proteins that actually coat the single strands, otherwise it would just snap right back together before anything could happen. Okay, because DNA wants to be, or needs to be, or just is, by chemistry, double-stranded, unless we physically force it to be apart. Yeah. Yeah. So the helicase doesn't go away. So that was one of the things that's wrong with that picture. As the fork continues to open, the helicase stays on all the way until it either falls off the end of the chromosome or bumps into another helicase coming from the other direction. So that was one of the, like, the protein can't go away and then it continues unzipping. It pretty much drives that the whole way. Exactly. So the start of replication is called the origin of replication, the beginning of replication. And it's a sequence, it's a double-stranded sequence that the initiator proteins, or the machinery, the molecular machinery that actually sits there, recognizes. Right? So it's specific double-stranded sequence, that's the origin of replication. The proteins are what sits on the origin and does the enzymatic job, but the actual origin is a double-stranded sequence of DNA. Okay? That's a good question. In prokaryotes, there's one single origin of replication. One that, whatever the sequence is, occurs once, because the bacterial chromosomes write a small little circle. In eukaryotes, we have lots of different origin of replications because the chromosomes are so big. Even our smallest chromosome is orders of magnitude larger than a bacterial chromosome, right? It's a circle. If we cut it and opened it up, it would be way, 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 way shorter than even our teeny tiniest little Y chromosome. Okay? So we need lots of origin, which we just abbreviate ORI, because otherwise replication would take forever. If we had one single origin in the middle of every chromosome, instead of replication taking 5 hours, 7 hours, whatever it takes, it might take 5 or 7 days. That's not physiologically useful to us if we can't mitose, one cell in under a week. So there's just multiples, and the machinery sort of sits down all at the same time, and replication starts happening all along the chromosome, right? Whereas here, this guy's small, there's one origin, and then replication goes this way, it meets somewhere in the middle, done. For the eucs, if this really is the end here, right, this goes all the way till it falls off. All the shit just falls off the end. This one goes this way, this one goes this way. When they bump into each other, then they fall apart, they're done. When these two bump into each other, everything falls off, it's done. This one either bumps into another one, or if it's the end, falls off of that end. Okay? So this is all sort of happening simultaneously. And it's really just to make it efficient by timeline. Okay, so the major difference between replication in prokaryotes and eukaryotes is one origin of replication for the prokes, and it's a circle, and multiple origin of replication on the chromosome. So the bigger the chromosome, the more origins, 1,000 of them on chromosome 1, right? Less on the smaller ones, but so that we can get our entire genome replicated in that S phase in 5, 7, whatever hours. Okay, so that's just for speed and efficiency. Yes? Yeah. So when the two bubbles meet, right, essentially this one bumps into a strand over here and this one bumps into the strand over there. Yeah, and then that first part, all that machinery falls off, and then part 2 of replication happens, like the little tutorial, when we have to pull out the primers and get everything back to one strand. We'll talk about that. Probably we'll get to that tomorrow. But yeah, they essentially, right, the two stick, and then all of that machinery falls off, and the new machinery to clean it up and make it all one continuous strand then hops on to all those spots. And we'll get to that molecularly. As long as we understand in the eucs, it's not just one spot. Otherwise, essentially, everything that happens on the molecular level is exactly the same between prokes and eucs. Eukaryotes are more complex. They have more enzymes doing different things. I'm going to simplify it in here, and we're going to use prokaryotic replication, the molecular stuff, and the names of the enzymes as replication. Eukaryotes, all of those proteins have a homolog, and then a few more that help, but we're not going to learn all the proteins involved in anything anyways, because it's crazy. Right? Every level that you step up in a course, you learn more proteins that are involved in all these things. But memorizing names of proteins in a complex that has 100 proteins is dumb. If you end up studying replication, you decide it's your thing, you're going to get a PhD, and you're working on the origin of replication, you're going to know all the proteins involved in sitting on the origin and what they do and their amino acids and where they contact DNA, and you're going to tell all your friends when you're drunk and just drive everybody insane. So we don't give a crap. We're going to call them initiation proteins. The huge complex targets the sequence at the origin to set up the bubble. And so they just call it the replication bubble because if you really look under a high-powered microscope, it literally is open, right? So this right here is that, this is that, and this, right? So the forks are moving that way. The origin was somewhere in the middle of that. Okay, so that, and the, ooh, look, it's a little bubble in the DNA, whatever. Okay. Yes, what I said here. Just like that Yahoo said, except the helicase enzyme, like I just answered, stays on and keeps breaking hydrogen bonds. Okay, so I'm going to try to use the real molecular terminology instead of unzip, and some books call it melting, which is really bad because it has nothing to do with heat, right? That's just a bad term. You can melt DNA, which is not really melting anyways, but you can break hydrogen bonds using heat, and that's how we do it when we do fake replication in a test tube. What's fake replication in a test tube called? Anybody? PCR, yeah, polymerase chain reaction. It's DNA replication in a test tube. Any molecular biology technique is based on something physiologically relevant. We take some DNA, we make our own primers, we put in DNA polymerase. We don't need helicase because we just heat the crap out of it. Remember, hydrogen bonds are not strong. They're strong in numbers, but individually they totally blow, right? So if you heat it to 95 degrees Celsius, almost boiling, right, they just fall apart. You have single-stranded DNA. As soon as it cools, right back together. So as it's cooling, if you have primers, they're little, they're 20 base pairs, they can stick at a much higher temperature than, right, the whole thing needs to be a lot lower. You can pop your primers in there, throw polymerase, boom, DNA replication goes. Okay, so all those techniques are based on something physiological. We just isolate all the enzymes we need, and we use heat because heat is more efficient than adding another enzyme. Right? You could just add helicase, but then every time you heated up the reaction, you'd have to add more helicase because heat kills enzymes. Doesn't kill that DNA polymerase because we use DNA polymerase from bacteria that lives in thermal vents at the bottom of the ocean. So they live in boiling water. So obviously if you survive boiling water, your enzymes must be able to survive boiling. And so it doesn't matter how many times we boil to separate the DNA, the polymerase is just happy-go-lucky to complementary base pair. Whereas our polymerase, if we boiled it, would done, dead. We'd have to add it every round, blah, blah. So helicase is stupid because we'd have to add it every time or isolate helicase from those thermal bacteria, and it's just easier to put in a machine and heat it up because that breaks hydrogen bonds. So I will call it the enzyme that breaks hydrogen bonds if I'm trying to give you a, backwards definition. Okay, that's its job, is to break hydrogen bonds. Where are the hydrogen bonds? Yeah. 2 between the A's and T's, 3 between the G's and C's. So it's harder to break through GC-rich DNA than AT-rich DNA, right? So if you're in a lab, blah, 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 and if your sequence is really GC-rich, then your temperatures need to be higher to deal with all this crap. Okay? Yeah. Hang on. So the other thing about DNA polymerases are that they cannot do complementary base pairing to naked DNA. So they can't just hop on this single strand of DNA and start complementary base pairing. They need an existing nucleotide sitting there attached already to the template to add to those ends. So we have to use the primase thing. So in the little video, they showed the big purple blob coming in and setting up what we call a primer, right? A primer is primary being first, start stuff, and it leaves that last nucleotide with its 3' OH flapping in the breeze. Remember how every nucleotide has a 5' end and a 3' end? Right, if I draw a really bad nucleotide that Dr. Stein would make fun of. Right, remember that's the 2' OH, and then here's your base. Remember we just, is it, oh, see how I am? Right, the phosphate group, which I'm just going to call a P, but we know it's like the oxygens and all that bullshit up there, right? This is the 5' phosphate, it's on the 5' carbon. This is the 3' OH, right? And then this would be where our hydrogen bonds are over there. So the last nucleotide added has this 3' OH dangling off, right? The 5' end is dangling on the other side of the primer. DNA polymerase needs this 3' OH to then attach to phosphodiester bond the next nucleotide. So that's why Dr. Stein, I hope, was jumping around about phosphodiester bonds. Those are between nucleotides, right? Phosphate sugar, OH, phosphate sugar, phosphate sugar, phosphate sugar, right? It creates the phosphate and the OH gets together in a condensation reaction. What comes out in a condensation reaction? Water. Okay? Makes that phosphodiester bond. That's between the nucleotides. They're between that primer. The primer's held to the template with the hydrogen bonds by complementary base pairing. That primer has to be anti-parallel, right? If we had the template 5', 3', and I say, there's our primer, and this line is just a bunch of nucleotides. I'm just lazy. I don't want to write A, G, G, C, C, A, T, G, blah, 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 right? I'm just being a lazy ass by drawing a line. Because at one point I had a student, like at the very end, ask me what those lines were. It's just a single strand of DNA. It's just all the nucleotides phosphodester bonded together. Right? So for that little primer on this side, which side is this? 5' or 3'? 3. Which side is this? Yes. Okay? Does that make sense? So then if this is the 3' OH hanging off, this would have the 5' phosphate hanging off. Which side would DNA polymerase grab and start complementary base pairing, make a phosphodester bond to? Yes! Those are like the best things ever. And so it would proceed in that direction. This side doesn't do diddly squat, it just sits there. Okay. So helicase, you need to know the name of that enzyme, the guy that breaks open the hydrogen bonds, opening the bubble. Yep. No, hold on to that. Leading and lagging both require a primer to begin. So we should write that down right now, so it's in your notes, so that you don't freak out. And books spend a shitload of time talking about Okazaki fragments and the lagging strand and the primer and blah, 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 but the leading strand must have a primer also, because DNA polymerase cannot start a new strand without an existing nucleotide there. Whether it's leading or lagging, cannot complementary base pair to naked DNA. Okay? Let's see what's next here. Okay, so this is a little more helicase, and then I'm going to write a little deal. All right. So helicase does the opening, and this is another thing I hate about textbooks, which when I write my own, then it'll all be good again. Yeah, right. Right? They're just showing you a snapshot. That's not necessarily the end of the chromosome. The other side, right, of the bubble's over here. They're just showing us how it's opening this way. So its helicase is breaking the hydrogen bonds. Remember, it's the curled up double helix. It's not just the straight two lines. So as we open this, as we break those, this stuff here gets all knotted up. Right? Because if you try to open something that's already twisted, it gets even more twisted. It used to be when dinosaurs roamed the earth and telephones had cords, you know, and there was one telephone in your house, Okay, I know you guys have no experience with that. And it's in the kitchen, and it's got the long-ass cord because you need to be able to walk into the bathroom with the phone. And it's a curled thing, and then that gets curled on itself, and then you're trying to walk, and it's curling because it's stuck at the bottom, and then it gets all knotted up, and then you pull it out from the wall, and then your mom's bitching and everybody's screaming, right? That's what happens to DNA. And so instead of mom coming and unwrapping it all and crabbing at you the whole time, we have an enzyme, class of enzymes called topoisomerase. It's a whole class of these kind of enzymes, one of which, I'm not sure if our book talks about it or not, but I think you may have heard the word DNA gyrase, is a type of topoisomerase. And its job is to break phosphodiester bonds, not the hydrogen bonds, actually clip a phosphodiester bond and let that supercoil loose and then seal it back up so that it eliminates that supercoiling. So otherwise, the fork would get stuck, it would be all knotted up, here you are, and you can't go any farther. Right? A bubble coming the other way would have knotted all this crap up, and you can't go anywhere. And that would stall replication. So if you were born with a mutation, or you couldn't be born, if there was a mutation in your topoisomerase genes and you can't reduce this negative supercoiling, your zygote, when it tries to go through replication, is going to stall, and if that happens, guess what? Dead. Dead happens, right? So we don't see individuals with a topoisomerase mutation because they never pass the zygote stage, because they can't do DNA replication, okay? So topoisomerase is your mom uncurling, or your grandma, great-grandma's house still has that phone, got the dial. Right? And that's what reduces that allows it to, and so that'll happen all along in front of the fork. Right? So I just scribbled all over the green guy. We'll draw him back in here. So this is the topoisomerase doing its job. It's following in front of helicase, right, going that way. From this bubble, there's a helicase and a topoisomerase, they're going that way. Right? Another helicase here, topoisomerase, they're going that way. Okay, so those are two enzymes we need to remember what their job is. And those questions will just be, what is their job, right? Or, you know, you see an enzyme that's breaking phosphodester bonds and then clipping them back together. It's in front of the helicase. What is it, right? Okay. Now what? Oh, so this is the other little guy that I talked about, right? Which we're never going to draw again on our bubbles because they just take up too much room. Remember we talked about just a few minutes ago, DNA cannot stay single-stranded because hydrogen bonding as a whole is very strong, pulling them back together. So when the initiator proteins sit and open and helicase starts breaking hydrogen bonds, single-stranded binding proteins need to bind all along to keep them apart. Okay? And so then when primase comes in to stick a primer, it will remove the single-stranded binding proteins to start complementary base pairing, remove them as they go, right? Remove them as they go. And so we're going to know that they exist, but we're not going to draw them all over our DNA, or we can't figure out what's going on. Because we know double-stranded DNA can't stay double-stranded unless we hold it, or something else is already in there. Okay. So what else do we have to do? We have to have the template, and the template is just the double-stranded DNA that's the chromosome that's sitting there, right? Chromosome 1. One of chromosome 1, the other chromosome 1. Both chromosome 2s, chromosome 3s, all of your DNA. And then again, the primer, which is, in our case, made out of RNA nucleotides, and it provides that juicy 3'OH for DNA polymerase to add. Okay? All enzymes, I'm going to make a statement that's close to lying. All enzymes that we're going to talk about that do complementary base pairing always, always, always add in the 3' direction. Add to 3' ends. Right? Start. Here's our template strand. Right? Our other one's somewhere else, but we're ignoring it. If we put one nucleotide, has to be anti-parallel, right? And the next nucleotide is going to be off the 3' OH. All polymerases, all enzymes that do complementary base pairing do this. So primase has to start with one nucleotide, pop it there. The next one would sit right there. This would be number 1, number 2, number 3, etc. Right? This is the 3 moving in the 3' direction. New strands always travel 5' to 3'. And when we talk about all these processes, whether it's replication, transcription, kind of translation, PCR, any of the other molecular techniques, we're always talking about the newly synthesized strand when we say replication proceeds 5' to 3'. The new strand. Because you can't say any polarity, which 5'3", 3 to 5, about an entire process because the template strand's always anti-parallel. Right? So when we're talking about a process proceeding, we're always talking about the new strand, not the template. Because the template's always opposite. Because it's anti-parallel, it has to be. So 5' to 3' always refers to the new strand. The primer's added 5' to 3'. The new strand grabs the 3' end of the primer. Complementary base pairing happens 5' to 3'. Yeah. The 3' end is the end with the OH. The 5' phosphate. So when you go back and look at those stupid nucleotides from chapter whatever, 3 or 4, I can never remember, right? Each nucleotide itself has a polarity. 5' up there, 3' OH, which means the whole strand has a polarity. And it's labeled by this end has the phosphate hanging off. That's the 5' end. This individual nucleotide is 5'3", 5'3", 5'3", 5'3", and then this one's what's hanging off, so that's the 3' end. Yeah.
So you're saying that it goes 5', 3' direction, but it adds on to the 3' end?
Yes. So each strand proceeds 5' to 3', and we're always adding to 3' ends. So if we, let's do this. Let's do this. And we have to upload something. And so we'll do this. 5' to 3'. And then we're going to, right, our other strand is down here. We're going to, right, because it's DNA. This is DNA. But we're going to ignore this one down here just because, we don't care about him right now. And we're going to do like I did before, and we're going to have a primer. And then you guys are just going to label it, the 5' and 3' end, and then draw an arrow which way polymerase would proceed. Okay? And then I'm going to erase this one down here just because we know he's there, but we're not going to think about him. And then while you guys are doing that, I'm going to set up another little... So here, do the top part. So just do the top part, screenshot and upload to whatever today's first thing is. I'm just setting up the bottom part for the next upload. So it takes me a long time to write stuff and I didn't have one set up. So screenshot, just do your top guy, screenshot and upload. Yeah, 2 separate ones. So just do the little easy top thing. Yeah, so just put the polarity 5', 3', and which way polymerase would extend, would grab, which end is it going to grab and start doing phosphodiester bonds and complementary base pairing? And then I'm going to tell you. Okay, and then you can, once you screenshot it and uploaded the easy top one, you can start drawing out the bottom one, and then I'll tell you what to do. Right, so the top one has to be anti-parallel, right? That means the phosphate's on that side and the 3' OH is there, and that means we'd extend off of the 3' end. Right? The rules of all these molecular genetics is has to be anti-parallel, right? Rule number 1. Has to complementary base pair. And we always add to 3' ends. So those are the 3 rules you need to live by for all of these processes that we're going to talk about. All of them. Nucleic acids, whether it's RNA or DNA, if they're going to complementary base pair, has to be anti-parallel. Has to be. Because if you look back at the stupid nucleotides, that's the only way they fit together for hydrogen bonding, right? Being upside down and backwards, right? To get the A and T to fit in the G and C. If they're both the same way, it doesn't work. Complementary base pairing is just ATs, GCs, right? Always, always, always adding to 3' ends. So you can't go the wrong way if you know those 3 rules. If I ask you to put if I tell you this is a template strand, yeah. It's the primer. Yeah, it's a great question. So if we're talking replication, the primer is RNA nucleotide. Right, so the U's are going to pair with the A's instead of the T's. So yeah, so we're going to say, this time we're going to, if we say this side is 3' of our template and this side is 5', right, add the polarity to our primer, and from here on out, all of this here is supposed to be DNA, because that's really our point, right? We're trying to make DNA. We're trying to take a DNA strand and make more DNA, but we have to start with these RNA nucleotides because that's what the primase enzyme is specific for. That's all it can add. It can't add DNA nucleotides. So it has to add RNA nucleotides. That's just what fits in its active site. Okay? And that does help with regulation, so there aren't mistakes and replication happening in bizarre places. Right? This primase can only use RNA nucleotides. It only gets expressed when the cells are getting the signal to go through G1S, G2, and start the cell cycle. So it has to do with regulation, and that's just probably how it started, and it just never changed. So that's a good, another question is, DNA nucleotides and RNA nucleotides complementary base paired as well together in a DNA-RNA hybrid as DNA-DNA or RNA-RNA. Right? Because if you think about, remember the base, the actual base part, the cytosine part, the guanine part, exactly the same between DNA and RNA. What's the difference between DNA and RNA nucleotides? It's just the sugar part, right? The base is exactly the same, except for the stupid U, right? But the U has the same 2 hydrogen bonding sites as a T. That's why he fits with the A. And T's just don't exist as a ribonucleotide. So the hydrogen bonding, as far as chemistry is concerned, doesn't know, doesn't care. Hydrogen bonding, DNA to RNA, DNA to DNA, RNA to RNA, doesn't give a shit as long as it's anti-parallel. Yeah. Why the cells use RNA? No, I don't. It's a good question. I don't know. Evolutionarily, we think the first nucleic acid was an RNA molecule, and so I can only imagine that if somehow during evolution, if you look at the structure of uracil and thymine, they're very similar. Some little change in chemistry happened, was more stable, or could just be frickin' random, right? So I don't know. I don't know enough of the biochem about